Good afternoon. It's really a great pleasure to introduce Scott Baraban, who's one of my favorite scientists and a really great friend here to UCLA in person. It's wonderful to see him and talk to him, and it's really great that he's meeting everybody, and well, it's rewarding to have everyone together again. Scott did his PhD at University of Virginia with Patrice Guyenne, with Lothman, studying sympathetic outflow and during opiate withdrawal and multiple other topics. It was a very productive PhD. And then he went on to do a postdoc with Schwarzkreuz and Palmater, studying CA1 neurons in models of prenatal cocaine administration and neural migration disorders, and then moving on to study the anti-epileptic effects of MPY. He eventually made it to UCSF in 1999 as faculty, and he spent a great deal of time studying these models of neural migration disorders, as well as studying NPY. He looked at potassium channels and studied synaptic dysfunction and cortical malformations from the MAM model to tuberous sclerosis models to LIS1. But then he, a little bit of divergence, where he discovered this, and he pioneered the use of these zebrafish models for studying epilepsy, which really allowed him to do genome-wide mutagenesis screens and later on large-scale drug screens, which he continues to today. He will speak about the zebrafish work tomorrow. He has 2 lectures on 2 separate topics, but the zebrafish work has been extremely productive and has given rise to the discovery of a number of candidate molecules and actually pre-approved drugs that have already been approved that can be tested in humans. They're already being tested, actually, he was telling me. And tomorrow he'll talk about those drugs, I think, in detail. He has the zebrafish models are models of Dravet and Angelman's and a number of other epilepsies. He also has moved into studying neural transplantation as a treatment for epilepsy. These are precursors to GABAergic neurons that are transplanted in multiple models. And this is what he's going to talk to us today about. The KV1.1 model, the temporal lobe epilepsy Pilo models, and the newest work is actually transplantation into the sea lion Cronut that everyone has been coming up to me saying, make sure that he talks about the sea lion. So as you can see from the picture, I hope he will talk about. So he is now the professor in William K. Bowes Jr. Endowed chair. In neurosurgery, he has won the Klingenstein, the Sandler Family Supporting Foundation Award, the UCSF Innovation Award in Basic Science. He's won Eureka grants from the NIH and at Javits, and multiple other awards. So please welcome Scott Baraban.
Thanks. I think I have this. I'm good. Great. Well, thank you for a very kind introduction. And it is actually amazing to finally be back on the road speaking semi-live. I know some people are tuning in, but just to see some faces here is a real pleasure, and old friends and hopefully some new friends as well. So today I wanted to talk to you about our interneuron-based projects. This is, as you'll see, a story that involves several species. This was working when I got here. So I'll give you an overview. For those of you not familiar with epilepsy, I'll briefly discuss in the introduction what epilepsy is and the types of epilepsies we're focusing on and the different types of projects in the laboratory. And most of the talk is focused obviously on interneurons, which is a specific class of inhibitory cell in our nervous system, and the medial ganglionic eminence, which is one of the regions where interneurons originate from. Most of our research over the last 15 to 20 years on this topic has involved rodent models and rodent MGE. But in the last 4 or 5 years, we started to work with MGE derived from pig fetuses. And I'll tell you a little bit about that. And then finally, the very photogenic and apparently more popular than I realized, epileptic California sea lion, in this case, named Cronut, who we've been working with the last year or two. So as Peyman introduced, the laboratory has 2 general themes in the last 10 to 15 years. One part of the lab is focused on genetic forms of epilepsy. And for this, we primarily work with zebrafish models that we've developed that mimic specific rare genetic epilepsy seen in children. And if you want to hear more about that, I'll be giving a grand round talk in neurology tomorrow morning about our zebrafish work. But today I want to talk to you about the MG projects. So these projects use embryonic neural progenitor cells. These are not stem cells. These are actual fetal tissue. They're not manipulated or grown in any way. And these cells are destined, as you'll see, to become inhibitory interneurons, actually specific classes of inhibitory interneurons. And we're using them to pursue novel cell-based therapies to treat intractable forms of epilepsy. And for this, we largely focus on the other spectrum of epilepsy disorders, which are acquired forms of epilepsies, normally seen in adults. And for those of you who are not familiar, epilepsy is a fairly common neurological condition. It's actually, I think, the 3rd or 4th most common CNS disorder. And it's marked by a very specific electrical event, which is shown here. This is an EEG trace on several channels of a patient during a spontaneous seizure. And so an epileptic condition is defined and marked by the sudden recurring episodes of these sensory disturbances, loss of consciousness, and usually motor convulsions, and they're associated with abnormal electrical activity. There are different forms of it. This is an ictal type of epileptic form event that's generalized. You see it in all the channels. And there's another electrical signature called absence epilepsies, which have more of a spike wave pattern. But the important point is epilepsy is defined as having 3 or more of these spontaneous events. They're not induced in people, and they can occur for different reasons, but they always have an electrical component. And that's helpful as we think about modeling them in the laboratory and preclinical models, is that we're able to record similar electrical events in simpler species. So epilepsy, like I said, is fairly common. 1 in 26 Americans actually will develop epilepsy at some point in their lifetime. It's most prevalent in children, and then again in the aged population. Worldwide, at least approximately 65 million people suffer with epilepsy. One out of 3 of these patients is medically refractory, meaning they don't respond to available approved treatments, and their seizures remain uncontrolled. And in children, it's one of the most common neurological disorders, particularly the genetic forms of epilepsy that occur early in development, and they're usually rare, devastating forms of this disorder. So what are the available treatments? Well, there are several options. None of them, again, is completely curative, but they're all helpful in controlling seizures in different patient populations. 2 of them involve stimulation, vagal nerve stimulation, or deep brain stimulation approaches to control some forms of seizures. In children, sometimes you can get seizure control with a form of a ketogenic diet. This is actually fairly effective in a number of pediatric epilepsies. And in some adult forms of epilepsy, specifically temporal lobe epilepsy, where there's damage to one of the hippocampus, one of your hippocampi, resective surgery has shown to be very effective. In fact, a lot of that pioneered right here at UCLA by the neurosurgeons and neurologists who've been doing temporal lobectomies for many decades now very effectively. And finally, anti-epileptic drugs. And these run a wide variety of mechanisms, from drugs that modify inhibition to drugs that affect ion channels and calcium channels, sodium channels, et cetera. And there are about 28 different FDA-approved anti-epileptic drugs. But again, 1/3 of all patients with any type of epilepsy don't respond well to any of these treatments. And so when I got involved with epilepsy research, and as you heard, took somewhat of a circuitous course, I guess, to studying epilepsy specifically. One of the early projects I was involved with had to do with neuropeptide Y, which is a transmitter found in inhibitory cells. and focus on inhibitory neurons that my mentor's lab, Phil Schwartzcoin, was doing a lot of work on in the late 70s and early 80s before I joined the lab. And this came from an idea that was actually somewhat simple-minded, in that when you look at tissue samples from patients with epilepsy, specifically temporal lobe epilepsy, and some of this work was done here by Carolyn Hauser and others, you see a loss of inhibitory cells, in addition to the other changes in sprouting of excitatory cells. And so this led to a hypothesis, which is actually in the early literature. This is actually from a quote in the 1969 Basic Mechanisms of the Epilepsy book. This is a meeting called the Jasper meeting that occurs roughly every 10 years in the epilepsy field. And this is a chapter in that book by Alden Spencer and Eric Kandel, who worked in epilepsy at the time. And it talked about surround inhibition, which is basically this idea that you have inhibitory cells surrounding an area or a focus of epileptogenic activity. And epilepsy, as stated here, is a common topographical picture of synaptic imbalance. This is simply how many of us think of epilepsy, whereby an excitatory pacemaker region is surrounded by a powerful ring of inhibition. And of course, we now know that inhibition to this region is provided by inhibitory cells. So it makes sense that when surround inhibition breaks down, you have more seizures and are prone to epileptogenesis. And so one of the early thoughts, simple-minded thoughts I always had, was what if we can restore or somehow enhance this area of surround inhibition? Would that be therapeutic? And so it's not, again, surprising that inhibitory cells or inhibition are important. Not only this early hypothesis, which was based on EEG recordings in rodents and people at the time, but a host of literature over the years that has shown that if you block inhibition, usually pharmacologically with things like GABA receptor blockers, whether you do this acutely in brain tissue or in vivo even, you can induce what looks like a seizure-like event. And that's shown here on the left. And more recently, work by Ivan Soltej's group, as well as others, have found using optogenetic techniques that if you stimulate these inhibitory cells using optogenetic approaches, in this case, parvalbumin specific interneurons, you could abrogate or stop these induced seizure events. And so there's a long history that inhibition is important in epilepsy. And experimental evidence, like I said, ranges from anatomical evidence of loss of these inhibitory cells through functional evidence that you block inhibition or reduce inhibition, you induce the seizures, to more recent evidence that you can stimulate existing inhibitory cells and stop a seizure. So all of this converges on one class of neuron in our brain. In a very broad sense of the billions of neurons we have, there are two broad classes: excitatory cells that release glutamate and drive excitatory synaptic transmission, and inhibitory cells that release the neurotransmitter GABA, which is an inhibitory transmitter that hyperpolarizes cells, so brings them further away from firing. So interneurons as a group, which represent about 20% of the total neurons in our brain, mediate all forms of inhibition. Interestingly, however, they are very heterogeneous. There's different flavors of interneurons, and the diversity of them is actually pretty significant. In the early days, I'd say when I was a graduate student, there were roughly thought to be three types of interneurons. But now we know there could be up to 15 or 20, depending on how you parse them. And they work in different places. So this schematic shows you a typical excitatory cell, a pyramidal cell releasing glutamate, and different types of inhibitory cells that impinge on this cell. So you can see some impinge at the soma. These are usually parvalbumin expressing interneurons that control excitability by inhibiting cells here, GABA receptors located here. Others are on the dendrites, and some even innervate other interneurons. And so there's a very heterogeneous class of interneurons. And some of these are therapeutic, and some of these, if you put them back in or activate them, would inhibit seizures, while others would actually make seizures worse. So to think about replacing interneurons, you first have to know, where do interneurons come from? Luckily for me, the way I got involved with this is through two excellent collaborators at UCSF who showed up in my office one day around 2000 or late 1999 and showed me pictures of inhibitory cells that they had transplanted. Those collaborators are John Rubinstein and Arturo Alvarez-Bulla. And what John had found, studying where interneurons come from, is that there was a specific region in the embryonic brain that was transient called the ganglionic eminences. And this is an E13.5 section of a mouse embryo. from these ganglionic eminence regions, progenitor cells that are destined to become inhibitory cells migrate out and eventually get to the neocortex and hippocampus, where they integrate as inhibitory cells. Separately of that, Arturo had found that the migratory properties of these cells were not just something you saw during development, but could be maintained in vitro. So if you take these cells out and culture them in matrigel, you see that they start to show these migratory properties. In fact, he had shown at that time when I met John and Arturo that if you take out labeled cells from the medial ganglionic eminence and you transplant them into another animal, they'll actually migrate and integrate in the host animal. And that's basically the basis for a lot of what I'm going to show you in the next few minutes. And finally, as this story has progressed over the years, and I'm forgetting a lot of contributors, but Gord Fischel's lab and Oscar Marin are key contributors to this. We now know a lot about the molecular profile of these cells that derive from the ganglionic eminences. And this is important, as you'll see, for the work we do, because it allows us to figure out what types of cells we've actually transplanted. And so there's a series of transcription factors that are present in different regions of the ganglionic eminences that define the different subpopulations of inhibitory cells that you're going to get. And as you can see from the diagram, it's quite complex, but there's a couple of key features that allow us to define different subpopulations of inhibitory neurons that come from different ganglionic eminences. So the first part of our journey to a therapy for epilepsy starts with the medial ganglionic eminence. And I want to, of course, highlight some of the very talented, at the time, postdocs, but now independent on their own right, people who contributed to this story. Bobby Hunt, who's a professor up the road at UC Irvine, Mackenzie Howard, who's at UT Austin, Mariana Casalio, who worked on the pig cells that you heard about, is back in Argentina, and Rosie Paterno, who's soon to leave the lab and start her independent lab at the University of Oklahoma. So our approach, of course, with the idea in mind that we want to put new inhibitory cells in epileptic animals, is to develop a transplantation protocol. So the way this works is we take embryos, in this case, I'm going to talk about mouse embryos, at E13.5, which is the peak of when this transient medial ganglionic eminence structure is developing in the embryonic brain. We dissect out, using anatomical landmarks, MGE progenitor cells. We then dissociate those cells and transplant them into another animal. There's no immunosuppression necessary. These are mouse-to-mouse studies. We see very little inflammation at the site of the injection. Without showing you all that data, basically what we know is that the needle itself, or this depositing the cells itself, elicits a little bit of an immunoreaction response, but days later, nothing. And the cells aren't rejected. It's mouse-to-mouse. No need for skid mice or anything like that. So we put these into host animals. We transplanted 3 different age groups, depending on the type of experiment we're doing. We can do this at pups from postnatal day 2 to 4, juvenile mice, or in the case of epileptic animals, often adult animals. And we put the cells into either cortex or hippocampus, depending on what the experiment is, and which of the area where we want, what area we need to increase around inhibition might be, given the type of disorder. And then we look at the animals different days after transplantation, or DAT, between initially 30 days, but all the way up to 2 years later. And the readouts we're looking for to see what happens to these cells, and again, they're labeled with GFP so we can track them in the host animal, is of course typical histological approaches using antibodies, staining, and things like that. Electrophysiology to see the functional integration of those cells. Behavioral readouts to see how they impact the whole animal at a behavioral level. And video EEG monitoring to see effects on epilepsy. And so there's a couple of features that we need to track to know we're looking at MGE-derived interneurons. So like I said, there's a medial ganglionic eminence, and that primarily produces somatostatin and parvalbumin expressing interneurons, which is a set subclass. There's a caudal ganglionic eminence, and that primarily produces VIP and real positive interneurons that actually make inhibitory connections with other inhibitory neurons. So they have a different transcription factor profile, but they also have a different function, which is very important. And through John's work, which is shown here from one of his many elegant anatomy studies looking at embryonic brains, we have a set of transcription factors that allows us to profile cells as being from the MGE versus the CGE. from Arturo's work, and this is some early work done with Arturo around 2005 or so, we found that if we transplant these cells, again, transplanting MG cells into a naive animal in cortex at P2, we found they have tremendous migratory properties in vivo. So 3 days after transplant, you can see them up to 2 millimeters away from the injection site. And at 60 days after transplant, up to 5 millimeters in both directions. So this basically allows us to populate the host brain with interneurons across the entire cortex. And if we go and look at what types of interneurons these cells become, they do indeed have the profile of MGE-derived interneurons. So here the green GFP-labeled cells are co-labeled with either somatostatin, parvalbumin, or nNOS, which are all indicative of an MGE-derived population. They do not co-express glutamatergic markers. They do not co-express astrocytic markers or microglial markers. And they, more importantly, don't co-express things like VIP and reelin and calretinin, which are CGE-derived markers. And interestingly, and this is important for the work we're actually kind of progressing on now, depending on where and what age you transplant these cells, the host brain actually has an influence on how effective this therapy or treatment could be. This is work that Rosie's working on at the moment. And what she's doing is comparing transplants into cortex, hippocampus or cortex, a single injection at P2, juvenile or adult. And what we're finding, which is fitting with how we can kind of tailor this to different types of epilepsies, is that the injections or the transplant works most effectively in young animals, not surprising. Development factors and other things that would favor integration of new cells is best at this age point. And it actually works better in cortex versus hippocampus in terms of the spread. And that's partly because there are some anatomical landmarks that turn out to limit the spread in the hippocampus, one being the dentate gyrus, where they migrate to the dentate gyrus, but then they stop there because they're not going to go past the granulocell layer. And so either way, whether you do hippocampal injections or cortical transplants and look at P2 up to adult, we can populate the host brain with many of these new inhibitory cells. It's a little more effective in cortex versus hippocampus, which is shown here, and it's a little more effective in P2 versus juvenile or adult animals in terms of the number of cells that survive and integrate. Most importantly, and this is really where I initially got involved with the project with Arturo and John, it's one thing to show green cells in the host brain. That doesn't mean they're doing anything functional. And it turns out if these cells aren't functionally integrating, they're not going to be effective in treating epilepsy or anything else. They're just going to be sitting there. And so one of the first things we did, and we repeated many times over the years, is to track these cells down, because they're GFP labeled, in slice preparations, either hippocampus or cortex, and do patch clamp recordings from those cells to see what their firing properties are. All of our neurons, of course, have different electrical fingerprints or firing properties in current clamp mode when you depolarize a cell or hyperpolarize a cell. Interneurons are, of course, fast spiking and have different waveforms that are indicative of the type of subclass they are. And if we record MG cells, either 30 or up to a year later, we find that they look like different classes of inhibitory interneurons as we'd expect. And so 5 of those are shown here: parvalbumin, somatostatin, et cetera. Furthermore, if we go and do single cell PCR, we suck out the contents of the cell after we've recorded from it, we can match up their molecular profile with their electrical profile. And indeed, somatostatin cells that have somatostatin firing properties match with expression of somatostatin. And here they also receive excitatory inputs from the host brain. There's a quick voltage clamp trace at the bottom to show that. And so the bottom line is when we recorded these MGE-derived interneurons in either cortex or hippocampus using patch clamp techniques, we only find cells that have inhibitory interneuron firing properties. And again, this fits with the anatomy. We didn't see glial-like cells or astrocytes, and we didn't see excitatory cells. So that shows that the cells can functionally mature and be active in the host brain, but it doesn't say they're actually having effect of functional integration into the circuit. It just says that their intrinsic firing properties are normal. So the second step is to look at host brain cells that presumably would receive synaptic inhibitory input from our transplanted cells. And we've done this a variety of different ways. I'm just going to briefly show you a couple here. One is to transplant cells, MG cells, that are labeled with a channel rhodopsin, expressing a channel rhodopsin. In this case, it's a parvalbumin driving a channel rhodopsin. That way, when we record slices from these animals, And when we look at the host brain neurons, the pyramidal cells that are presumably receiving input from inhibitory cells in the circuit, and we stimulate these cells, we can show that they actually functionally integrate and inhibit pyramidal cells. So if we optogenically activate PARV cells, we see an inhibitory response on the host cell, again, blocked by gabazine. And these responses look identical to what we get if we just looked at a native PARV cell that we stimulate. So they seem to functionally integrate and they make synapses. This is parvalbumin, but we've also done this with somatostatin. And so they make either soma innervating synapses or dendritic innervating synapses that are inhibitory. Another way to do this, to show their direct functional integration, is to simultaneously patch clamp two cells and to see if they talk to each other. These are very labor-intensive studies, and these were done by Mackenzie Howard when he was a postdoc. And again, looking just at the parvalbumin experiment, this shows you if we record a native pyramidal cell and an MG-derived parvalbumin interneuron, and we see if they talk to each other, they do indeed talk to each other as we'd expect. And so this suggests that they're making synapses that are functionally integrated and not just kind of sitting there and firing on their own. And most importantly, and this is really the critical feature of how this differentiates from a lot of what I consider stem cell derived approaches that show a neuron is firing, but don't show an effect on inhibition. So again, the initial premise for all this is that we're trying to increase surround inhibition and therefore constrain an excitatory focus or ictogenic focus. And so for that to happen, we would have to be able to increase inhibition by putting in more interneurons. And indeed, when we do patch clamp experiments, again, either hippocampus or cortex, looking now in regions where we see GFP-labeled interneurons recording host pyramidal cells, we've consistently, over now almost a 15-year period, in many different postdocs in the lab, focus on looking at synaptic inhibition. So these are voltage clamp recordings, looking at inhibitory postsynaptic currents. And as you see, these little traces, this is a typical trace. This is an inhibitory iPSC. These are blocked by GABA receptor antagonists. If you look at grafted animals, starting around 28 days after transplantation, this is important because before that, we still see cells with migratory properties, so they haven't yet matured. And before that, when we did intrinsic property recordings, they weren't yet mature either. So this fits with the idea that functionally mature cells are integrated. We see an enhancement of the overall level of inhibition onto the excitatory cells. This is usually in the range of 20 to 30%. And again, we've seen this with 5 different postdocs at different times in the laboratory, so it's at least internally replicated. And we've seen this both in cortex, hippocampus, adult, young, however we do the transplants. What's interesting is we can't overexpress or overstimulate. So when we put in more cells, like 500 times more cells, we might see more cell somas, but we don't see an increase in inhibition over 20%. So it doesn't overscale. Basically, the way we think of it is the host brain has a certain amount of real estate that could allow new inhibitory inputs to come onto, but once they're there, there's a threshold for that. So this is fortuitous from a therapeutic point of view because we can't put in too many inhibitory cells and basically sedate the animal. And so it goes up to about 20, 30%, but it doesn't increase linearly as you put more cells in.
I have a quick question. When you were characterizing the transplanted cells with electrophysiology, and you were looking at cortex, for example, have you ever seen if they receive direct thalamic connections, like they made it one foot?
Yeah, so we haven't done thalamocortical. So it was asked whether when we look at cortically integrated transplants and doing electrophysiology, whether we've assayed them to see if they receive thalamic input as would be expected for cortical interneurons. We don't do thalamocortical slices, so I don't know the answer to that in vitro. But I do want to point out, which is on these slides, and again, this summarizes a lot of electrophysiology that went on for probably a decade before I got to the epilepsy therapy stuff. And so if I'm skipping something, feel free to ask a question. We also looked at inhibitory synapses, so or synapses onto inhibitory neurons. So iPSCs on presumptive inhibitory cells, and we saw no increase in the level of inhibition onto inhibitory cells. And again, that fits with the selective integration of these cells as PARV somatostatin subtypes that are going to inhibit excitatory cells. So we don't boost the level of inhibition in these animals with the grafts. More recently, and this is really just a flavor of kind of where we're going next with this. We've done a lot of work, like I said, in vitro to show functional integration, and now we're trying to move in vivo. As you can imagine, these are experimentally harder and involve a lot of different techniques that are more modern and slower to get up and running, but they're all kind of fascinating. One is to just use simply 32-channel silicone probes and look at oscillations at the local field potential level. As many know, oscillatory activity is driven by different types of interneurons, and this allows us in a direct way to functionally see how we're influencing oscillatory activity that's driven by inhibitory cells in animals where we put in more inhibitory cells. And so that's one approach we're taking. This is just some examples from Rosie's work, looking at theta rhythms, which are regulated by a type of interneuron. We're now upping that a little bit by adding single unit recordings onto these probes. So we have simultaneous LFP recordings and single unit recordings, which allow us, of course, to isolate putative inhibitory cells and excitatory cells during these different cycles. And again, integrating that onto animals that are receiving transplanted cells. And then working with Matt Strayman at UCSD and hopefully Peyman here at UCLA, we're going to start to look in vivo at calcium imaging and hopefully voltage imaging as a readout of what these inhibitory cells are doing in a whole animal after we transplanted them. And this is some 2-photon images from Matt's lab, very preliminary data, obviously, just to show, as we're getting this technique up and running, that hopefully we can monitor the activity of inhibitory neurons in live animals after transplantation. So with all that in place, and again, that was work for the students at least, and postdocs to get some perspective, that probably was about a decade in the making to get to the point where we wanted to transplant these cells into animals to see if they were therapeutic. And summarizing some of that work, and I just want to briefly touch on it because these are all long stories, but they're now have been in literature for many years. Taking these cells, we then transplanted them into various animal models of epilepsy, rodent models. And so, like I said at the beginning, there are genetic forms of epilepsy based on usually a single gene mutation. Often these are childhood epilepsies, and the patient has epilepsy throughout their life, lifelong epilepsy. And these can be modeled, of course, in rodents by knocking out that same gene. So we wanted to test MG cells in what are called genetic forms of epilepsy. And then on the other spectrum, you'll hear more about acquired forms of epilepsy. And to briefly summarize all of that, the initial paper in 2009, we found if we put MG cells into the cortex of a potassium channel knockout mouse, we could reduce seizures by over 90%. And when I say reduce seizures, these are video EEG recordings 24/7 for 10 to 14 days of a live, freely moving animal. And we're only looking at seizures that are at least 15 to 20 seconds long. So these are that have a convulsive behavior and have a long duration. So these aren't epileptiform events. These aren't spikes or anything like that. These are full-blown seizures the animal have spontaneously. And so in KV1.1 mice, we saw a 96% reduction in the number of these spontaneous seizures. In a DLX1 knockout mouse, which is related to an interneuron deficit from John Rubenstein's lab, we reversed a gamma oscillation deficit. Again, interneuron mediated. These mice don't have very many seizures. And more importantly, in acquired forms of epilepsy. So these are traditional models. You take a wild type animal, you give it pilocarpine or some other drug to induce status, and then later some of those animals survive and develop full-blown spontaneous seizures. In this model, which is widely used and is pharmacoresistant, that is, many labs have tried to give AEDs to these animals with no success, we found an 86% seizure reduction at 2 months following transplantation in our first study in 2013. A couple years later, we came back and did similar studies, but looked at 6 months after transplant, and again found an 88% seizure reduction. And more interestingly, even in the seizures, through a battery of rodent tests that you're familiar with, Morris water maze, open field, et cetera, we were able to rescue many of the behavioral and cognitive comorbidities these mice also have by transplanting MG cells. I'll briefly take you through some of this data. So this is an example, like I said, of our video EEG monitoring approach. This is a typical electrographic seizure associated with a full-blown kind of convulsive behavior. The mouse falls over. It's 15 seconds or longer. All these video EEG monitoring was done in freely behaving animals, and all the analysis was done blinded by students or postdocs who were not aware of what the animal was in terms of treatment groups. And so if we count up these frequencies, pilocarpine animals, roughly 2 seizures per day. Control animals, of course, don't have any seizures. If we transplanted cells into the hippocampus, we saw almost complete elimination of these spontaneous events. If we put the cells in the amygdala, and there's reasons we did this, having to do with some literature that thought the amygdala was controlling these seizures, we see no change. And if you look at the anatomy, interestingly enough, we could put lots of cells in, but these, which are pilocarpine damaged animals and already have cell loss and mossy fiber sprouting, shown here in red, we don't reverse the underlying anatomical changes. So it's important to point out that in these animals, or even the KB 1.1 animals, we're not correcting the reason why they have epilepsy. We're just allowing, by increasing surround inhibition, the animals not to actually have seizure events by constraining these events. So that's 60 days after transplantation. This is a separate study by Mariana at 180 days after transplantation, a similar protocol, video EEG recordings, shown here what the spectrogram looks like. This is a 2-week recording, a histogram showing you a pilocarpine vehicle-treated animal compared to an MG control. Again, very few spontaneous seizures. And again, the cumulative data looks like the 60-day data, almost complete suppression of seizure events. Again, fitting with the idea that we didn't change mossy fiber sprouting or any of the underlying damage. If there was a seizure event, it had similar duration in both animals. So again, we're not correcting the reason why they have epilepsy, we're just stopping them from having seizures and the results of their epilepsy. And finally, very briefly, and this is a good way of showing it. If you just look at the animal, this is a pilocarpine animal with MG transplants at about 60 days after transplant. He just looks healthier and happier and brighter eyes and fur, et cetera. This is a pilocarpine treated animal, which is many of you know epileptic animals. Their grooming isn't great and their appearance starts to deteriorate as well as their health. And if you look at the data on an open field assay, which is considered an anxiety assay, we again can rescue these behaviors with pilocarpine animals that receive MG cells. And again, this is one of a battery of behavioral tests we run on these animals, just as an example. And finally, because it is important to point out what we did change, and we changed what we did fix, we were able to fix inhibition. Again, the entire premise is that by putting in inhibitory cells that make synapses and are functionally integrated, we would be able to enhance inhibition in animals that have reduced inhibition, which is an animal with epilepsy. And indeed, if you look at slices from these animals, these are the pilocarpine treated animals at 240 days after transplant. So they've already been video EEG monitored or behaviorally monitored, and they went to slice experiments. Pilocarpine treated animals, as noted and shown here by Istvan Modi and other groups, Paul Buckmaster, have a reduction in iPSC frequency, consistent with him having a reduction in the numbers of inhibitory cells. Pilocarpine plus MG animals, again in the hippocampus, see a nice restoration of inhibition back to baseline levels. And so we believe this is the underlying mechanism of action for why these MG transplants work. Basically, we're boosting inhibition, synaptic mediated, GABA mediated inhibition. Because we don't think this is necessarily restricted just to epilepsy, and this is a very new part of the lab, so I really just want to show this one slide, but I encourage people to read this recent paper, which should say 2021, by Rosie Paterno in Cell Reports. We're now branching out into other types of disorders where inhibition is effective. As many of you know, it's not restricted to epilepsy. There's a spectrum of disorders where reduced or altered inhibition causes neurological disorders. Autism is on that spectrum. Autism, epilepsy is on that spectrum. And so to get into that kind of field and to see whether the MG transplants have a more broad appeal to other conditions, we've started to look at a catnap2 mouse, characterized here at UCLA initially as having fewer parvalbumin interneurons. They had reduced inhibition as well in our hands when we looked at iPSC frequencies. And then we've been transplanting them, or first we started to characterize them using these LFP recordings to show the types of inhibitory deficits that we could pick up with this LFP approach, as well as the behavior studies. And so this is examples of changes in spike wave ripples, but there's also data in this paper about different oscillations and other deficits that are interneuron related. And this sets up a number of outcome measures in an epilepsy autism model. Again, they don't have very many seizures, so it's mostly an autism model to see if transplanting MG cells into their hippocampus can restore some of these deficits as well. So that's all well and good. And if you're a mouse and you come to my lab, I'm happy to treat you and stop you from having epilepsy, but not too many mice show up for that. And it's not really why we do any of this in the first place. Our real goal, obviously, is to get to larger species and hopefully to humans. But that's not straightforward, and you have to have a route to get there. So our approach, which contrasts from stem cell approaches, and not to knock stem cell approaches, because I think they're fascinating, but in the field of inhibitory cells or interneurons, the types of inhibitory cells we want to put in the host brain, which are pavoalbumin and somatostatin, are not routinely possible to generate from any iPS-induced pluripotent stem cell type of technology. And so we've always pursued an alternate approach, which is just to scale up our embryonic MGE project. And the way we've done that and the way I've thought about that is to focus on a porcine source or a pig source of MG cells. And the reason we've done this is twofold. One, of course, is that in the 1960s, pig valves were the first heart valve transplants before we had synthetic or artificial valves put into humans. So there's a precedent for using pig organs or tissue in humans. And more recently, some of you might have seen, there's actually at the University of Maryland, a group that raised a pig and harvested his heart and put that entire heart into a patient. And so it's the first pig heart to human xenotransplant. So with this in mind, we've been pursuing a xenotransplantation protocol where we would harvest MG cells from pigs rather than rodents. As you can see here, these are just examples of the pig embryos at embryonic day 35. We obtain them either from the vets at the University of California, Davis, or the swine center, which is located at the University of Missouri. So that's our source for fetal tissue. And I think I'm probably, at least as of a month ago, not alone now in thinking that these porcine to human transplantations could change the way we approach some of our therapies for intractable types of diseases. In fact, this quote comes from Dr. Cooper, Harvard Medical School, a little bit after the Maryland group published the pig heart transplant. And he had said, as you see here, I think this is the beginning of a complete revolution in transplantation. I think we concur. So before we could transplant larger species, we had to, of course, show or start to demonstrate that porcine MGE has the same characteristics of mouse MGE that we've been working with for over a decade. And so that starts with just looking at the embryonic tissue. This is just a nice sample, E35, stained with different transcription factors, as you'll see, of a pig brain. And so we started where we think John Rubinstein and Arturo had started, with the same types of experiments that they came to me with in 1999. The first, of course, is our knowledge of what transcription factors are expressed in MGE cells, MG regions. Again, work largely by John's lab. And so this now is a porcine MGE section stained with DLX2, which is one of the transcription factors that are specific for MG cells, and KX 2.1 as well, LHX6 here also, and not so much COOP-TF2, a little bit. OLIG2 is also seen there, and a little bit of GSH2. And again, these are all transcription factor profiles that mimic what John and others had shown to be present in the embryonic MGE in mice. And so they seem to be recapitulated. I'm just showing you a few here. The paper from Mariana, which came out last year in Neuroscience, has a full explanation of a lot of other transcription factors and beautiful pictures as well. Secondly, like Arturo's work, we wanted to see if they maintain their migratory properties in vitro. So we cultured the cells in the same type of matrigel system that he had shown in 1999 with Enoch Wichterle to demonstrate the migratory properties of MG cells. And it's important, which I didn't mention in Arturo's early work, that was very novel at the time because other types of cells, for other progenitor cells, don't have that migratory property. So if you look at progenitor cells, for example, from the LGE, which is lateral ganglionic eminence, they don't migrate in vitro. Or if you look at progenitors for either astrocytes or primal cells, they don't have this migratory property. And so it's an important feature of MG cells, both in vitro, but obviously in vivo, which allows us to do the transplants. So again, the cells can migrate in all directions. And if you do some simple staining types of approaches, you can see that you get inhibitory cells that co-express things like somatostatin and VGAT, et cetera. And so they have the profile in vitro of MGE progenitor cells. But most importantly, of course, we need to xenotransplant. So in these studies, we took the porcine cells, we transplanted them into adult rats, because we wanted a slightly bigger brain structure in which to analyze them. We did a brief immunosuppression protocol, because this was a xenotransplant, unlike the mouse-to-mouse studies. And then we looked at the animals between 7 and 60 days after transplant. Again, you can see that the cells migrate and start to integrate, and they kind of plateaus. In fact, we went out to 120 and 240, it looks similar. And more importantly, if you look at these pig cells, which are labeled with GFP, you can see that they start to integrate throughout the hippocampus, and they've migrated different regions, and they have the profile of inhibitory neurons. All of this mimicking what we saw with mouse studies. And when we did immunohistochemistry to profile them, we showed that the GFP positive cells co-express GABA, somatostatin, and TUJ1. Unfortunately, we could not find a parvalbumin antibody that worked to differentiate pig cells transplanted into a rat. Sadly, we tried about 4 or 5 different antibodies, but we couldn't get any convincing staining, so I don't have data on that. And another interesting observation that finally came out of all this is that the pig MG cells seem to sit somewhere between mouse and human. So Mercedes Paredes, who helped us with some of this work and had a beautiful study recently about human MGE and some of the features of the human MGE, including these dividing neuroblasts, these little pockets of cells. Notice that while she was doing this work, parallel to what we were doing in the porcine brain, that we were seeing the same types of dividing neuroblasts in the porcine MGE that she was seeing in the human MGE. And again, this was a science paper recently by Mercedes, and it's important to point out it was interesting, not only because it's a beautiful characterization of MG cells in the human, but also because this was never seen in rodent or rodent MGE in any of the studies. So the porcine cells not only represent or replicate many of the features we know about MGE, they also seem closer to human than they do to mice. So that's all well and good. We've got preclinical evidence that we can put in inhibitory cells, transplant them, they'll become interneurons of a certain subclass. It's worth pointing out that in those therapeutic studies where we showed rescue of epilepsy treatments in mouse models of epilepsy, when we put in CGE-derived interneurons, so those that will make VIP or calretinin inhibitory cells that will innervate other interneurons, we not only didn't reduce the seizures, we actually couldn't make wild-type animals have epilepsy. And so interneurons that innervate other interneurons are not therapeutic. In addition to preclinical evidence that MG cells seem exciting as a potential therapy, we also know that we have a source now to do larger species. This source is the embryonic pig brain. But how do we get proof of principle in a larger animal that would get us close enough to consider doing a patient one day? And so we thought about this for a long time. And what always struck me is that because it's so invasive to make rodent models of epilepsy that I'm talking about. So pilocarpine, for example, 20% of the animals we inject with pilocarpine are going to survive. So for every 10 animals, we're going to lose 8 before we even get an animal that we could do a transplant on. And the frequency and the duration and the severity really varies in those types of acquired models. And so there's no way to ethically think that, oh, we're going to do that to a larger animal. We're going to give pilocarpine to a monkey or whatever. And so there's really not that path. If you think of genetic models, it's even more complicated. First of all, many genetic models of epilepsy, KB1.1 mice I talked about, die early. Those mice actually die around 45 days of age. And so you really can't do a lot of long-term studies on them. And again, you'd have to genetically modify a larger animal, even to get an epilepsy model. So we were kind of thinking like, well, where will we test this? And it struck me, and this is work really that I've seen for many years living in California, about the sea lions. So as some of you know, we have sea lions here on the West Coast that during harmful algal blooms that occur in the summers, they ingest a harmful amount of domoic acid from these algae blooms. Domoic acid is an excitotoxin. It's similar to canic acid. These animals, and they've been studied for 2 decades now, develop hippocampal sclerosis, interneuron loss, and adult onset epilepsy. And they show up on the beaches of California where they get collected by the marine mammal centers for rehabilitation and release. And what they've noticed over the years is that these animals have a very high morbidity. Over 75% of them don't survive when they're released. They also have difficulties in navigation, not surprising for hippocampal damage. When their trackers are put on them, many of them will go from San Francisco to Seattle for the winter instead of down here to San Diego. They have atypical aggressive behaviors and cognitive impairments and a host of other comorbidities. And most importantly, they're a naturally occurring form of epilepsy. And a lot of this comes from a colleague of mine, Stanford, very talented colleague, Paul Buckmaster, who's been studying the brains of these sea lions for about a decade. And so Paul's published some very beautiful anatomical studies and come to some very profound conclusions, which is basically that these domoic acid poisoned sea lions have the same type of synaptic reorganization, rewiring, and interneuron loss that people with temporal epilepsy have. So this is actually a naturally occurring form of TLE that mimics what we see in humans. 80% of the time, the damage is just on one side, it's unilateral, again, like patients. And the synaptic organization, as you'll see, and the loss of interneurons are very similar. And it's from this work that Paul had presented at many epilepsy meetings over there that kind of inspired me to see if we can do something for these sea lions. And so this is an example of what Paul is talking about. This is a brain section of a domoic acid poisoned sea lion. You can see the atrophied hippocampus here on the right. And again, this resembles what you see with temporal lobe epilepsy in patients that come to the clinic. Again, acquired form of epilepsy. TLE is the most common form of adult epilepsy in the world. And this photos come courtesy of the Marine Mammal Center. And as I mentioned, Paul's work from Paul Buckmaster's lab, looking at these sections in more detail, he found that they had hippocampal atrophy as noted, but also a significant loss of parvalbumin expressing interneurons. Again, this is what we'd expect in an animal with epilepsy and mimics patients with TLE as well. In fact, this anatomy, when you put it side by side with human TLE studies, I think in one of my other talks, I think I one of Carolyn's slides actually, and slides from UCLA, you can see a very striking similarity between human TLE and the TLE that you see in these domoic acid poisoned sea lions. And finally, a group down at UC Santa Cruz, which has also been studying these animals, have found that they have spatial memory deficits. These are hippocampal dependent tests that they run them on. They've done both MRI imaging types of studies as well as whole animal studies, high resolution imaging to go with that. They teach them different tasks of, again, domoic acid poisoned epileptic sea lions that have been rescued. So this is kind of where we came in about 5 or 6 years ago. I had started to talk to the Marine Mammal Center in Sausalito in Northern California about these sea lions and pitching them on a plan I was hoping we would have to treat a sea lion with these porcine cells. We weren't really sure how it was going to go, and we spent a lot of time at the beginning just working on the logistics, as you can imagine, of like how we were going to do transplant in a sea lion in the first place and things like that. And then on, as you'll see in a little bit, September last year, I heard about Cronut. So Cronut, who many of you know probably through social media, if you follow this kind of thing, is a sea lion living at Six Flags Discovery Park in Vallejo in Northern California. Cronut originally stranded here in San Luis Obispo. He's about 3 years old. In 2017, he was found to be depressed. I'm not sure how they analyzed that, and lethargic. He was moderately underweight, and he was suspected of having domoic acid poisoning. He was released, treated and released. This happened 2 more times. And eventually he was adopted, or he was picked up at the Marine Mammal Center in Sausalito. They confirmed domoic acid toxicosis. They observed a seizure. That was in April of 2018. And in June of 2018, he was adopted to live full-time at Six Flags Discovery Kingdom in Vallejo. MRI, as part of a project funded by Ben Inglis and others at UC Berkeley on these animals, was performed at that time, and they found mild atrophy of his left hippocampus. About a year later, in 2019, a second and third seizure were observed. He was started on AEDs, phenobarbital and diazepam. And then in September of 2021, he was having multiple seizures, severe weight loss, and he was actually designated for euthanasia. And so this is where I met Sean Johnson and Claire Simeon, the vets who were taking care of him at Six Flags. This is the behavioral decline of Cronut after he was adopted at Six Flags. And this is basically tracking how much, what percentage of his weight he's losing. Sorry, what percentage of food he's eating each day. You can see he was fine through 2020 and then had a couple of dips. Now in the summer of 2020, he went almost a month without eating anything, no matter how they presented it to him. And so I received an email on September 1st of 2020. Turned out Sean and Claire had seen me give a talk at the Marine Mammal Center about this porcine MGE to sea lion approach I was had. And they said, hey, we have this sea lion. He's in severe condition. He's been having many seizures, a horrible weight loss, and we're about to euthanize him. Do you think you can come out and take a look at him? And not that I'm a sea lion vet at all. I drove out. Turns out that's not far from where I live. And I met Cronut and we discussed a plan. And that plan, which happened at the beginning of COVID, was to scramble together a team to see if we can do a pig transplant on Cronut before he died. And so because it involves many people, and this is just a few of them from that day, I just want to kind of acknowledge them up front. It took about 20 different people came together for this, veterinarians, his handlers, Brain Lab, Laura from Brain Lab, which is an MRI group, the Marine Mammal Centers, et cetera. And the plan then was to see if we can transplant pig cells into Cronut's hippocampus. And the approach was, of course, to use these interneurons and a stereotactic approach to target the hippocampus that was damaged on one side. We had already, like I said, been working with the Marine Mammal Center for the past couple of years to develop a customized needle that would allow us to transplant the cells into a sea lion brain. And we've been playing around with different techniques and stereotactic approaches that we had worked out. These, of course, are all modified for a sea lion skull and their anatomy and scaled up, of course, from rodents. And with the approach to inject directly into the temporal lobe. And our hope was to combine this with MRI-guided stereotactic approaches. That's where Brain Lab came in. Basically, we're going to get MRIs and CTs on site and then deliver the cells based on those. And so this is a little bit of the background of how we got there. A couple years earlier, we went to the California Academy of Sciences down the road from me, and we measured different sea lion skulls to get us an idea of the landmarks. Those of you who do stereotactic surgery know we need some fiduciary landmarks. We, of course, knew the anatomy from Paul's work. And again, we had an MRI on Cronut himself that was taken by Benningless at Berkeley about a year earlier. So based on all these, we decided on a trajectory to target the damaged hippocampus and deliver cells. This is a little bit of the pictures from that day. I want to emphasize again, this was during COVID. So of all the people in the picture, maybe 20 of us were there. Only 5 people were allowed inside the vet clinic at any given time that afternoon. And so we rotated people in and out to get this done. Here's Mariana from my lab. She's preparing the cells and the needle for injection. This is John Andrews, one of the neurosurgery residents. And this is the frame, of course, adapted from humans to fit on Cronut, and this is, of course, Cronut anesthetized right after his MRI. And so we had a customized needle, again, to deliver the cells. We showed up with our pig progenitor cells, which were frozen. We targeted the left hippocampus and enteral cortex, 4 different locations along the track, and relatively small number of cells, 50,000 cells per injection site. But amazingly, it all went well. We were there for about, I think, 6 or 7 hours. Cronut came down on a crate from Vallejo, was anesthetized, intubated in the parking lot, wheeled inside, got his MRI, all that stuff, and got his transplant. Went back in his crate, drove back to Vallejo. This is Cronut the next morning in his octopus pool at his apartment at Six Flags. They kept him dry docked for 7 days after transplant, because we had drilled a small hole, of course, to do the injection. He's remained on a low dose of AEDs, phenobarb and diazepam. Again, similar to what you do with patients who have temporal lobectomy. We don't want to withdraw these drugs because that could lead to a withdrawal-induced seizure. He was initially on steroids for inflammatory reasons, and he started eating really the next day. We gave him some appetite stimulants. There were very few complications, some swelling around the pin site and where we put the frame on, largely because the frame wasn't made for a sea lion, so we had a little trouble getting it set. His temperature was elevated, some mild inflammation. He was a little bit lethargic, and he also remained on some analgesic and supportive care. And now we're actually, as of this morning, so first we're 17 months post-transplant, and Cronut is doing amazingly well. It's the only way to describe it. Here's a picture of Cronut at, I think, 16 months. This is his weight. You can see it was precipitously low, about 25% of it he lost before the transplant. He's been back to normal since then. He's gone over a year completely seizure-free. And he's not only having his seizures controlled, which again were the week before we had him transplant him, he had 12 seizures that weekend. So this is not a trivial change in his seizure frequency. And in addition to his weight, Cronut's actually starting now at about 12 months to look like what his trainers say is a normal sea lion. And he's actually, for the first time since they've had him at Six Flags, they're able to start to train him to be in the sea lion show. And I want to show you a little bit of that training here. Yes, so he stays on he's on a low dose of phenobarb and diazepam. Again, we don't want to, after the video held up. He's on a low dose. We talked to, I emailed about 7 different neurologist friends of mine, and the consensus was if he's fine, don't leave him, don't change it. Amy Brooks-Cahal actually said the main reason she takes her TLE patients who have resective surgery off of AEDs is because they want to drive. A reason that Cronut probably won't want to drive, so we're going to leave them. Them also being, one of the drugs being phenobarb, we're worried about withdrawal-induced seizure at this point because he's been on it for a while. So yes, he stays on a low dose of both. And there's longer video of him, and actually, you know, last month when I was out there, he's able to now not only follow his right and left flipper, but he can go from one trainer to another, learn different directions. And again, these are all things that Cronut never could do before the transplant procedure. And so to summarize, what I hope I showed you and convinced you is that embryonic MGE progenitors are a source for a subpopulation of therapeutically interesting GABAergic interneurons, that these MGE progenitors can migrate, they can differentiate, and most importantly, they can functionally integrate following transplantation. That if we transplant mouse MG cells into mouse model of epilepsies, they're curative. And if we transplant pig MG cells, xenotransplant into a domoic acid poisoned sea lion, it's also curative. And so this gives us hope that we can now maybe think of a path that would get us to the clinic for this type of approach. And like all things, I want to thank, of course, NIH, which has funded this for many years. My laboratory, some of them here shown, the recent turnover, and all the current members of the laboratory working on this project and many others. And I want to, of course, thank those of you who showed up in person and those of you online that I can't see. Thanks for coming out as well.